This is the 8th video on the fundamentals of biology, and today we're going to be talking about the cell cycle. So here's some basic terminology that we need to know. A somatic cell is all cells in the body that exclude the sex cells, also known as gametes, and they undergo mitosis. All somatic cells will undergo mitosis. And mitosis is when one parent cell forms two identical daughter cells. The genome is all the DNA inside of a cell, and a chromosome is packaged DNA. There are a total of 46 chromosomes in a somatic cell, with 23 from each parent. A chromosome is made up of chromatin, which is DNA and protein, and it condenses into an X shape after replication and during division. So this X shape will be a chromosome that is duplicated. And sister chromatids are duplicated chromosomes during division and are attached at a centromere. So this first thing would be a sister chromatid, and when it's duplicated, it forms two sister chromatids, but is still considered one chromosome. And in the middle here is a centromere. For 90% of a cell's life, somatic cells are in interphase and are not actively dividing. So in this circle, you can see that most of its life is spent within interphase. The first step of interphase is the G1 phase, also known as first gap, and this is where the cell simply grows. The next phase, called synthesis, is when DNA is duplicated and is used to be split into two cells later on in life, as shown in this step. In the G2 phase, the cell will continue to grow and ultimately prepare for mitosis, which is the next step in the cell cycle. The M phase, also known as the mitotic phase, consists first of mitosis. The first step of mitosis is called prophase. In prophase, the chromosomes will condense, the mitotic spindle will form, and the nucleolus will disappear, which is originally the site of RNA synthesis. So what is a mitotic spindle? A spindle is created by the centrosomes, which is a pair of centrioles. So in this diagram, two centrioles will make up one centrosome, and a centrosome is duplicated in interphase. So there will be two total centrosomes. And they are made up of microtubules, and they will attach to chromosomes at kinetochores found at the centromere. And non-kinetochore microtubules are called astromicrotubules, and they help to elongate the cell. So in this diagram, the kinetochore is found in the middle of a chromosome and is attached by a microtubule, whereas the ones that are not attached to any chromosomes are called astromicrotubules. In prometaphase, which is sometimes also considered part of prophase, the nuclear envelope will break down and release chromosomes, and the spindle will continue to grow. In metaphase, all 46 chromosomes will line up at the metaphase plate, which is in the middle of the cell. The next step of mitosis is called anaphase, and this is where sister chromatids will separate into chromosome pairs, and they will go to the opposite ends through things called motor proteins. And you can see in this diagram, sister chromatids are separating from 46 total chromosomes to 92 total chromosomes. However, the number of sister chromatids will stay the same since they already duplicated in prophase. And finally, in telophase, the spindle will ultimately break down, the nuclei will form again between the two cells, and the chromosomes will decondense. So telophase is basically the opposite of prophase, except it will happen between two cells. The final step of the mitotic phase is called cytokinesis, and this is when the cytoplasm is finally divided to create two new cells. In animals, this process is called cleavage, and it will create a cleavage furrow in this middle. And in plants, a cell plate will form. In the cell cycle, there are checkpoints, which are cues to determine whether it is ready or not to continue with the process of mitosis or the cell cycle. So between the G1 and the S phase, there's a G1 checkpoint, and this basically determines whether or not the cell will actually divide. And things that it will check for is if the cell is large enough, if it has enough nutrients, if there are growth factors that will actually tell it to divide, and it will make sure that there is no DNA damage. If these conditions are not met, it will enter something called the G0 phase, and cell division will not occur. This is prevalent in things such as neurons. The next checkpoint happens between the G2 and the M phase, and this makes sure that no DNA is incorrectly coded. It will make sure that all the DNA is replicated and undamaged, and will wait for repairs if it is damaged, but if it's not possible, apoptosis will occur. Apoptosis is programmed cell death. Finally, between metaphase and anaphase, there will be the spindle checkpoint. It will prepare DNA to be divided between two cells. It will check if all chromosomes are attached to spindle fibers, and if this does not happen, it will wait until all chromosomes are attached at the kinetochores. Cell checkpoints happen through the use of cell cycle regulators, and these help cell division to either stop or continue. An example of these are called cyclin. Cyclins are proteins that promote the activity within each phase. So within each phase of the cell cycle, such as the G1 phase and the S phase and the G2 phase, there will be the growth of certain cyclins. And cyclin-dependent kinases, or CDKs, are enzymes that are activated by a cyclin and is then able to modify target proteins by phosphorylation. An example of a cyclin-dependent kinase is the maturation promoting factor, or the MPF. In this, the M cyclin will bind to CDK, which induces the breakdown of the nuclear membrane. The APCC is another example of a cell cycle regulator. In the first step, it will add a protein tag called ubiquitin to the protein securin, and it will recycle it. And as a result, securin will no longer be able to inactivate the enzyme called separase. Separase usually will chop up the protein cohesin that keeps sister chromatids together. And as a result of separase not able to work, sister chromatids will separate, and the cell will be able to re-enter G1 again after the M phase is completed. Finally, P53 is a protein that is a tumor suppressor, and this produces a CDK inhibitor called CKI in order to buy time for DNA repair, and it activates DNA repair enzymes. If it's not fixable, apoptosis will occur, and this will prevent mutations from occurring. By having programmed cell death, it will make sure that infections are not spread throughout the body. Finally, let's talk about cancer. So what is cancer? This is when cells ignore signals that tell them to stop dividing, or do not undergo apoptosis, programmed cell death, when it should. How does this work? A change in the DNA code called mutations cause unregulated cell division, and proto-oncogenes will become oncogenes, which will stimulate growth even without growth factors. And proto-oncogenes are genes that cause normal cell growth, and once they become oncogenes, they will become cells capable of dividing too fast. The result of this is that a massive cell will build up on a certain part of the body. If this is unharmful, it is called a benign tumor, but if it invades other body parts and becomes deadly, it becomes a malignant tumor and causes cancer. So thanks for watching this video. This video covered interphase and cell division, cell cycle checkpoints, and cell cycle regulation.